Hello everyone, welcome to JS Core. I'm Rajvardhan and today we are going to discuss UPSC's previous year questions for prelims examination under an initiative Prelims Skilling Through Previous Year Questions, PYQs. And the subject that we are going to discuss is Science and Technology. And under Science and Technology, we would be discussing about a theme, and that theme is biotechnology, concepts and applications. Because if you go through previous year questions of UPSC, especially post COVID-19, you would find that most of the questions were related to biotechnology. At least 2 to 3 questions are there from this particular topic, and that's why it is an important topic for the prelims examination, right? So what is its importance? Let us try to understand the importance of this particular theme. So first of all, biotechnology weightage has increased in the prelims examination. 2 to 3 questions from this topic itself. It is a topic under Science and Technology, and from this topic itself 2 to 3 questions are being asked by UPSC every year, right? Now, in order to cover this particular theme, or in order to cover this particular topic, there are 2 important aspects that you should be aware of. First is about concepts. You should be aware of the concepts related to biotechnology, basic concepts related to biotechnology. For example, what exactly is genetic engineering, what is gene therapy, what are stem cells, what are different types of antibodies, what are different types of cells that are produced if an antigen enters in our body, right? Then you should also be aware of let's say different applications. Second is different applications of biotechnology. And if we talk about applications, your focus should be current developments. Right? Why these 2 are the important aspects of biotechnology? Because if you go through previous year questions, you will find that questions are information based questions. So your information base should be as broad as possible, right? Because the use of elimination technique is very restricted, especially in 2023 we have seen that elimination was not working, and even before 2023 the use of elimination technique especially for biotechnology is very limited, and that's why you should be aware of these 2 aspects, concepts and applications, because questions that are asked from this topic are more information oriented questions. Hardly you would be able to put, let's say, some kind of logical reasoning to use in order to solve questions, right? In 1 or 2 occasions you would be able to do that, but it is not the case that all questions from biotechnology you would be able to solve with the logical reasoning, elimination, etc. And that's why focus should be on information, right? So let us try to discuss the questions that have been asked by UPSC from this particular theme. So firstly, we'll be discussing about questions from 2023's prelims examination. First question is, Wolbachia method is sometimes talked about with reference to which of the following, right? So this was 2023 prelims examination question, and this question, or this Wolbachia method, was in news in 2021, and it is pure current affairs or current development related to biotechnology. And to be more precise, this current affairs is information based, because if you see the nature or the type of question that has been asked, it is a one-liner question. If you know it, you know it. If you do not know it, it would be very difficult for you to solve it, right? So Wolbachia method is a method wherein what we are trying to do, we are trying to make sure that the vector-borne diseases, mosquito-borne diseases, its spread is restricted. And this we are trying to ensure by putting one bacteria in male mosquito, and that bacteria is called as Wolbachia, right? So in Wolbachia bacteria, it does not allow the growth of virus, and as a result of that, spread of viral diseases due to mosquitoes we would be able to restrict, right? So that's why answer of this question is A. But here focus is not on answer. Our focus should be on what is the nature of question, what is the type of question. So the nature of question is inclined towards current affairs and the type of question is information based. So you should be aware of these kind of current developments related to biotechnology, right? So that is first. So this is first question, pure current affairs and information is the focus. Now next question is aerial metagenomics best refers to which one of the following situations, right? Now here you can apply some logical reasoning, right? You can apply some let's say mind in order to solve this question, provided you are aware of the basic concepts of biotechnology. Now metagenomics is one of the basic terms that you must have came across in biotechnology. When you study biotechnology, you come across various terms like genetic sequencing, gene editing, gene engineering, right? And another term that you may come across is metagenomics, or if not metagenomics, genomics, right? Genomics is a data and metagenomics is data about that genetic data, information about that genetic data, right? And there are number of ways to collect this genetic information, and one of the ways is to sample genetic samples or to collect genetic samples from the atmosphere. In air, in the form of aerosol particles, we have DNA samples, RNA samples or genetic samples of various organisms. And when we collect those genetic samples, that type of collection of genetic information is called as aerial metagenomics, right? So here it is possible for you to apply your understanding of the terms like genomics, metagenomics in order to solve this question, right? So this question can be solved based upon your understanding of the basic concepts in biotechnology. So here answer is collecting DNA samples from air in a habitat at one go, right? So from air, when you collect samples, that type of sample collection in order to study those samples is called as aerial metagenomics, right? For example, whales you must have seen, in order to take air, they come out of water, and as they come out of water, they lead to, let's say, spraying of let's say aerosol particles in air, right? As they exhale air, from that exhaled air there are various aerosol particles that are added to the atmosphere, and when we collect those samples, that is what is aerial metagenomics, right? So that is the second question, pure concept, and if you are aware of this concept, you would have applied that concept in order to solve this question, right? So that is second question. Answer of this question is A. Next, now this question was relatively, let's say, on the difficult side. It was difficult to solve this particular question because often when you read biotechnology, you might not come across these kind of terms, microsatellite. So question is, microsatellite DNA is used in the case of which one of the following, right? So it is slightly difficult question because these terms are there in the core, let's say, biotechnology, right? Those students who have done their graduation in biotechnology might be aware of these kind of things, otherwise this question was difficult, right? So now here it is pure concept, right? So on DNA, there are some sequences or base pairs of the nucleotides which repeat after certain let's say after every certain time or space, they repeat. Those types of base pairs on DNA are called as microsatellites. Generally, they do not lead to exhibition of any characteristics, right? They do not lead to any characteristic. That means you can consider them as blank genes, you can consider them, right? So these microsatellite DNA are the base pairs that repeat after every certain time on the DNA, right. So, this is pure concept. It is there is no, let us say, any logical reasoning that you would be able to put in this particular question. So, what is the use of this particular method? So, generally microsatellite DNAs, they are used to understand the prodigy, they are used to understand the characteristics or the pattern of inheritance, evolution, and that is why answer of this question is studying the evolutionary relationships among various species of fauna, right. So, other aspects, if you are able to, let us say, use elimination, then it is possible for you to solve this question. For example, stimulating stem cells to transform into diverse functional tissues. Now, this is not related to stem cells. Microsatellite DNA is not related to stem cells. If you are aware of stem cells, its concept, you would be able to eliminate this particular statement, right. Then promoting clonal propagation of horticulture plants. Now, again in cloning, hardly we play with DNA, right, hardly we play with DNA. We take somatic cell, we use let us say the nucleus of that somatic cell in order to get clone. Now, hardly we are tweaking with DNA, and that is why even you can eliminate this particular option C. And D is assessing the efficacy of drugs by conducting series of drug trials in population. Now, in order to understand the efficacy of drugs, till today we are not focusing on their impact on different genes or different, let us say, people with genetic makeup, right. So, that kind of thing we have not started as of now. There is a term called as pharmacogenomics. In that pharmacogenomics we would do that, but as of now we are not doing it. So, if you are able to apply some elimination technique, which is very difficult, you could have solved this question, right. But otherwise this question was difficult question, fine. So, what is the nature of this question? Nature of this question is again information, and this information is not related to any application, it is related to concepts part, right. So, it is related to the concepts part. So, this question, answer of this question is A, right, relatively difficult question. Next. Now, this question is let us say relatively easy question, right. Let us consider the question. Question is, consider the following statements. First, some microorganisms can grow in environments with temperature above the boiling point of water, right. So, here you can apply logical reasoning. Why? Because it is not saying all microorganisms, it is only saying that some microorganisms, right. In all three statements, the statements are not saying most of the microorganisms or all microorganisms. It is saying that some microorganisms can tolerate very high temperatures, some microorganisms can tolerate very low temperature, and some microorganisms can tolerate extremely acidic conditions, right. So, there can be these kind of microorganisms, right. These are called as extremophiles, right. The microorganisms that can tolerate extreme conditions are called as extremophiles. So, first is some microorganisms can grow in environments with temperature above the boiling point of water, right. So, it is a possibility that there would be certain microorganisms which would be able to grow even above boiling point temperature of water, right, and that is why first statement is correct. These microorganisms that can grow above the boiling point of water are called as thermophiles. First is correct. Second, some microorganisms can grow in environments with temperature below the freezing point of water, right. So, there are some microorganisms which can grow even below the freezing point, right, even they can grow in sub-zero temperatures. So, that is also a correct statement, right. Next is those microorganisms are called as psychrophiles. Those who can grow in extremely let us say low temperatures are called as psychrophiles. Third is some microorganisms can grow in highly acidic environment with a pH below 3, right. So, that is also a correct statement. Acidophiles are the microbes which can tolerate pH value between 1 to 5, right. So, the question is asking pH below 3, and that is a possibility. right? So all three statements are correct statements, and that's why answer of this question is all three. Now here you could have applied logical reasoning. If you are let's say if you have read about biotechnology, about microbes, then you could have applied that understanding in order to solve questions, right? So all three statements are correct, and that's why answer is C. Right. Next now we are moving into questions of prelims 2023, prelims 2022, right? So the questions that we have discussed were related to prelims 2022. Sorry, 23. Now we are starting with 22's prelims examination questions related to biotechnology. Question is, consider the following statements in respect of probiotics. Now again, concept, right? There are two terms, probiotics and prebiotics. You might have heard about prebiotics, right? So prebiotics acts as a food for the gut bacteria that we have, right? So it is a food that promotes, it provides, let's say, culture for the growth of gut bacteria. And in probiotics, what we do, we add or they act as food or supplements that consist of good microbes, right? They themselves consist of good microbes. Live microbes are there in probiotics, fine? So this concept you should be aware of. Right, the difference between prebiotics and probiotics. So first statement is, probiotics are made of both bacteria and yeast, right? So this is correct statement, because we have lactobacillus bacteria and we have Saccharomyces boulardii, right? Saccharomyces boulardii is the yeast that helps in the growth of let's say, that helps in the let's say correction of gut health. Our gut health is corrected by these microbes. It can be in the form of bacteria, lactobacillus for example, or it can also be in the form of yeast, right? Saccharomyces boulardii. These are the types of bacteria and yeast which can be used for the development of probiotics, which themselves consist of live microbes. And that's why first statement is a correct statement. Second, the organisms in probiotics are found in food we ingest, but they do not naturally occur in our gut, right? So that is not the case. We take supplements. These are the supplements, right? So why we take supplements? Because the gut bacteria that are there, they get a boost, right? So it's not that they are not naturally occurring in our gut. They do naturally occur in our gut, but when we take probiotic food, we make sure that it acts as a supplement for our body. And that's why this statement, which says that they do not live naturally in our gut, is incorrect statement. Fine. So second statement is incorrect. Third, probiotics help in the digestion of milk sugars. Right. So that is a correct statement. There are some let's say bacteria that help in the digestion of milk sugar. For example, lactoacidophilus bacteria help in the digestion of milk sugar. So third statement is a correct statement. Fine. So this answer of this question is 1 and 3, that is mentioned in option C. Second statement is incorrect. Now here it is more about the understanding of the concept. Right. So two parts, first is concept, second is application. This question is more of conceptual question. Fine. So that is about this question. Next now this question you should have expected, right? It was more expected in prelims 2021, but they asked it in prelims 2022, but it was an expected question. Question is on vaccines that we have used in India, and also they asked about Sputnik V. So let us consider the question. So this question you can put in the category of current developments, right? So it is, vaccines are a kind of application of biotechnology, and this application is was in use during 21 and 2022. Let us consider the question. In the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. First, the Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform, right? So this is again the question that was expected and more of current oriented, and you must have read about these things, that the Covishield is a vaccine developed by Serum Institute of India, and it was widely used in India as well as it was exported to other countries, and here it was a viral vector vaccine, right? Chimpanzee adenovirus was used to, let's say, build immunity against COVID-19, and that's why it was not mRNA, right? mRNA vaccines were later, means in 2022 and 2023, these vaccines were approved in India, right? So for example, vaccines that were used in the USA by Pfizer, Moderna, those were mRNA vaccines, not this Covishield. Fine. So first statement, that's why, is incorrect statement, right? Now if first statement is incorrect, you get your answer, right? So that was the possibility in 2022's prelims examination, not in 2023, and we don't know whether it will be applicable in 24 or not. But it seems that UPSC might not go back to these kind of questions, because if you eliminate one, you can get the answer very easily. So first statement is an incorrect statement. Why? Because Serum's vaccine, Serum Institute of India's vaccine, was not mRNA platform vaccine, it was viral vector vaccine where adenovirus, which leads to cold in chimpanzees, was used. Fine. So first statement is incorrect. Second, Sputnik V vaccine is manufactured using vector based platform. Correct statement. Sputnik V was vector based vaccine. Third, Covaxin. So Covaxin is another vaccine that was used in India, and it was manufactured by Bharat Biotech. Covaxin is an inactivated pathogen based vaccine. So this statement is also a correct statement, because in Covaxin, COVID-19 virus or SARS-CoV-2 was used for the development of vaccine. That means a virus that led to COVID-19 disease in its inactivated form was used in Covaxin, and that's why it was inactivated pathogen based vaccine. So second and third statements are correct, and that's why answer of this question is B, right? It was on expected line related to current developments of biotechnology. Fine. So answer of this question is B. Next. Now again, this was also on the expected lines, because due to the use of vaccines, what kind of response does our body generate? There were a number of articles related to the cells that are produced inside our body in order to build immunity, right? And those cells are B lymphocytes and T lymphocytes, and question is on them only. So it was again, so see here what happened, these were in news, so they were a part of current development, but it is not a current topic, right? It is a conceptual topic. So what they did, they got inspiration from current affairs and they asked a question on static part, right, or conceptual part. Fine. So question is, which one of the following statements best describe the role of B cells and T cells in the human body, right? So we know that B cells and T cells, or B lymphocytes and T lymphocytes, they play an important role in our immunity. B lymphocytes or B cells produce antibodies or proteins that help us take care of the antigens, and T lymphocytes or T cells, they are the cells that attack cells infected by antigen, right? So that is the role of these two cells. B cells take care of antigen itself, T cells on the other hand take care of cells infected by antigen. Fine. So correct answer of this question is they protect the body from diseases caused by pathogens. Correct answer is D, right? So here the pattern of question is very important, because you can study like that when you prepare for 24th examination, for example. So here what they have done, the topic which was in news, they have got inspiration from that topic, and the concepts, that means the static part related to the topic in news was asked. So this pattern you should follow when you study for this particular subject or any other subject, where current developments are hinting towards the topics from static portion which are important from the exam's perspective, right. So that is what is the pattern that you should try to follow. So answer of this question is D. So to sum up our discussion for this particular theme on biotechnology, we can say that the questions are of two types. First, conceptual information, and second, application based information, right. The questions are based on information, right. More of information based questions are there, right, not factual ones. So these concepts and current affairs you should be aware of, right, and try to study those concepts more, or try to focus more on those concepts which are in news, like this question. Fine. So that is how you can prepare for this particular part. Now you can solve questions related to the current affairs which are given in the link in the description box. So that is about our discussion for today. Thank you.